Raskow and colleagues initially sequenced the genome of an isolate of the E. coli strain that had caused the German outbreak, which was characterized by an unusually high incidence of hemolytic uremic syndrome, or HUS. The investigators then sequenced the genomes of 7 different isolates of enteroaggregative E. coli known to cause diarrhea, but not HUS. To visualize regions of difference between these strains, the 8 genomes were compared with 2 closely related genomes. In the first analysis, the investigators mapped each of these genomes onto the genome of TY2482, an isolate from the German outbreak that was recently sequenced and assembled by the Beijing Genomics Institute and placed in the public domain. Red indicates regions present in the TY2482 genome and absent in the corresponding comparator strains. Comparing the patterns across the genomes divulges regions specific to C227-11 that may contain genes pertinent to the pathology. For example, it is clear that the phage-like element containing the genes encoding HUS-causing Shiga-toxin 2 is common to C227-11 and TY2482, but is absent in the genomes of the other 7 comparator strains. The presence, indicated here in solid blue, of a different phage-like element in the genome of the outbreak strain, and also of most of the other genomes, suggests that this element is unlikely to be relevant to the severe pathogenesis of the outbreak strain. In a second analysis, the investigators mapped the genomes of each of the sequenced E. coli strains to the genome of the strain 55989, which causes diarrhea but is not known to cause HUS. Red indicates regions present in the 55989 genome and absent in the comparator genomes. Comparison across the genomes suggests that the relative absence of sequence in the outbreak strain is unlikely to be relevant to the severe pathogenesis of the outbreak strain.